in this unit, we're going to be learning about everything that you need to know for Unit 2. And please make sure if you have any questions to comment down below. Come join our community on Skool.com to find more additional resources. Active transport and gradients. Okay. Now, active transport can build gradients or maintain a concentration gradient. So it can either build a gradient that there's a low concentration on the inside, or it maintains that there is a low concentration on the inside. This means a cell can keep more of a substance on one side of the membrane than the other. So the cell can then work towards having a high concentration on the outside, okay, the membrane, and a low concentration on the inside. And it can maintain that. But to do that, we need ATP inputs. And because we use ATP, the membrane protein would change shape, the ions would move, and the gradient will be maintained. So let's see. Here I have ATP that is now binding to this protein, okay? Once it binds, okay, the shape will change. So this protein shape changes and it opens up to the outside and it releases those molecules. Now, once it opens up to the outside, these potassium ions can then come in, and once the phosphate is removed, it will change its shape back to downward facing, and it will release those potassium ions, and then it will accept 3 sodium ions again, and will pay the price with ATP. It will change the shape, it will release it to the outside, so every time ATP binds, the shape changes. Okay. Now, ion gradients and membrane potential. So the ions are unevenly distributed across the membrane, because remember, we are constantly trying to move these sodium ions out, and we move them from a low concentration to a high concentration. So we're paying the price with ATP. Okay, now because there's more sodium ions on the outside, it is slightly more positive. Okay, and the inside becomes slightly more negative because of this uneven ion distribution. This leads to a charge difference, and this gives us the membrane potential. Okay, this charge difference, this outside is positive and the inside is negative. This is the membrane potential. So what is an electrochemical gradient? It has 2 parts, okay? It has a chemical gradient, which is a difference in concentration, okay? So this is the sodium ions. They would be a high concentration on the outside. This is the membrane. And the concentration of the sodium inside would be low. This is a chemical gradient. Now, because sodium is a charged molecule, okay, it has a charge, it creates the electrical gradient. So we need the chemical gradient to create the electrical gradient, because this is the difference in charge. If there are more sodium ions outside, okay, this becomes slightly more positive outside, and on the inside, we will have more potassium ions, okay? So on the inside, it will be more negative, okay? Because the cell is actively taking these sodium ions and pumping them out, okay? It's keeping the sodium ions out. So because it's moving the sodium ions to form a chemical gradient, it creates this electrical gradients. So let's look at an example. We have the sodium potassium pump. Now, the sodium, okay, and potassium pump is also called the sodium potassium ATPase. Now, ATPase just means that this is an enzyme that can bind with ATP, okay? Because that ASE means enzyme. So it's an enzyme that works with ATP. Now, if we look at this pump, okay? So using active transport, we are constantly, let's use a blue color here. We are constantly pumping out sodium ions, okay? Now we are pumping out those sodium ions. Every time we pump out sodium ions, okay, the shape changes. So we pump out sodium ions, the shape changes so that potassium ions can now bind to this protein. And when this phosphate that changes the shape is released, the shape changes back. So then the potassium ions are released back into the cell. Okay. And because it maintains a higher concentration of sodium on the outside, it becomes slightly more positively charged. And on the inside, it becomes slightly negatively charged. So it creates this charge difference or this membrane potential. Now, the ATPase connection. So ATPase refers to proteins or enzymes that use ATP because ASE means enzymes. And ATP is just enzymes that use ATP. Okay. Now, it helps provide energy for active transport. So if ATP will bind to this protein, okay, the phosphate group would release and it will attach to that protein. And this releases that energy from ATP. Okay. Now, ATP hydrolysis. This is ATP hydrolysis. Hydrolysis, meaning we cut that ATP with water to release ADP and the phosphate, but that phosphate would attach itself to the protein first, okay? And this gives out that energy to move ions across the membrane. If you enjoyed today's lesson, please don't forget to like and subscribe and come join us on the Skool.com community page where you'll find AP Bio blueprints. And if you take a look inside, you will find that Unit 1 is completely free. See you there.